Good afternoon. Thank you for coming. It's a pleasure for me today to introduce Bárbara Olmeda as the coordinator of the Grupo Biomembranas of the SEBBM. And today she's going to introduce us her invited speaker. Thank you, Bárbara.
Thank you, Patricia, for organizing these webinars. Welcome everybody to this webinar organized by the Biomembranes group. And today, let me introduce Teresa Giráldez. Teresa Giráldez holds a degree and a PhD in biochemistry from the University of Oviedo. After various postdoctoral stays in the USA at several universities, such as Yale, among others, she obtained a Ramón y Cajal Fellowship at the University of La Laguna, where she is currently a professor. Professor Giráldez is a very well-recognized expert in the ion channels field. In this field, she has been working for over 25 years, including the study of their structure-function relationships and also in the regulation in physiology and in disease. And in concrete today, Teresa is going to talk us about BK channels and their role in neuronal synaptic plasticity. So thank you, Teresa. Thank you very much for accepting this invitation.
Thank you. Thank you very much, Bárbara and Patricia, for organizing these webinars. And it is an honor to be part of this webinar series. And I especially thank Bárbara Olmeda for this invitation and el Grupo de Biomembranas de la Sociedad Española de Bioquímica y Biología Molecular. It is really very special for me to be giving here this talk today. So first of all, let me briefly introduce the work from my laboratory. As it has been mentioned, we are based at the Institute of Biomedical Technologies at the University of La Laguna in Tenerife, in Spain. And here we study ion channels, mainly voltage-gated potassium channels involved in the control of neuronal function. We have worked, as you can see, with several types of ion channels involved in different pathologies, addressing the structure-function relationship of these channels and scaling up to animal models to understand disease mechanisms. And today I would like to share part of the work from our lab related to the function of large conductance voltage and calcium-gated potassium channels, also known as BK. These channels are expressed at the plasma membrane as tetramers of alpha subunits. The transmembrane regions of these subunits resembles that of voltage-gated potassium channels, containing conserved residues that act as voltage sensors and pore regions. And then in addition, a large cytoplasmic domain regulates calcium sensing by specific calcium binding sites that are contained within this region. Structure function studies, to which our lab has also contributed, have boosted up the elucidation of full channeled cryo-EM structures. But I am not going to focus on this part of our work today. Of relevance to what I'm going to be telling you about today, BK channels in physiological conditions, when they activate, they produce a large outward potassium current. And these may contribute to hyperpolarized membrane potential. BK channels are activated synergistically by voltage and calcium, as I said before, in such a way that the GV curve, the voltage dependence of activation of the channel, the GV curve is shifted to the left towards more hyperpolarized potentials when calcium is increased. And this is something, this property of the channel is something that can be used to estimate how much calcium is in the vicinity of the BK channel, as I will show you later during the talk. In most physiological settings, though, BK channels are activated by micromolar calcium concentrations, and this requires the close association with calcium sources. For example, in the pre-synapse, it has been proposed that BK channels associate to voltage-gated calcium channels to form so-called calcium nanodomains. When the action potential arrives, depolarization activates calcium channels, driving calcium in, and coincidentally activating BK channels, leading potassium out. The resulting hyperpolarization acts as a negative feedback on the calcium signal. Although this association of BK with voltage-gated calcium channel has been extensively studied, interesting questions related to this mechanism are still open and are actually growing, including what is the calcium concentration in these nanodomains? How are the dynamics of these complexes? What proteins are involved in their function and their regulation? And additionally, new calcium sources other than voltage-gated calcium channels have been defined and identified. And we want to know what are their potential new physiological roles. In our lab, we are working on some of these questions. In addition to understanding the structure function of the channels in the physiological context, we are actually working on characterizing and studying the association of not only with voltage-gated calcium channels, but also with NMDA receptors at the postsynapse. And this is what I'm going to tell you about today. So NMDA receptors are potential calcium sources for BK. Why? Well, NMDA receptors are tetrameric ion channels that have high glutamate sensitivity. The apparent EC50 is within the micromolar range. They are voltage dependent. They show voltage dependent block by magnesium, slow gating kinetics, and high permeability to calcium. NMDAR tetramers are formed by the compulsory GLUN1 subunits and then two GLUN2 subunits, of which there are several different types. And these subunits determine the deactivation constant and the ion flow. In this talk, I am going to refer to NMDAR containing isoforms 2A and 2B, which are predominant in the adult central nervous system. Of importance here is that NMDAR are coincidence detector of presynaptic and postsynaptic activity. The presynaptic activity provides the agonist glutamate, and the depolarization that is driven by activation of the AMPA receptor at the postsynapse causes the removal of the magnesium block, and then calcium influx through these channels is fundamental in several forms of synaptic plasticity. How does this work? Well, the proposed involvement, although I don't have time here to go through all these schemes with detail, but the proposed involvement of NMDAR in plasticity is shown in this cartoon. According to this hypothesis, the increase in membrane abundance of AMPA receptors associated to long-term potentiation, or LTP, is mediated by calcium and calcium-associated proteins signaling pathways that involve CAM kinase 2. And without entering in details, consistent with this idea, any signal that is going to blunt, alter, regulate the calcium signal mediated by NMDAR is going to abrogate, alter, modulate the associated LTP. So could BK act as a brake for calcium signals in this physiological context? Well, more than 20 years ago, Isaacson and Murphy showed that in the olfactory bulb, BK can associate with NMDAR in functional complexes that regulates neuronal excitability. So in excised patches from granular cells, addition of glutamate produced an inward current, immediately followed by an outward current. The outward component is inhibited, as you can see here, by paxillin, and which is a very selective inhibitor of BK channels. And both components are absent when NMDARs are inhibited specifically by a specific inhibitor, which is called MK801. Similar observations were more recently observed and reported in dentite gyrus in the hippocampus, showing very similar, as you can see, current recordings. And actually, convincing data was shown indicating that this association may be related to synaptic function and plasticity. And in fact, co-IP from brain extracts indicated that these associations may occur in different areas on the brain, not being restricted to this studied here. With this antecedent, in our lab, we decided to explore the role of NMDAR-BK complexes in neuronal plasticity by studying the barrel cortex region. The barrel cortex processes signals derived from peripheral somatosensory receptors. Within this area, pyramidal neurons from layer 5 have been shown to connect with thalamic afferents and are known to initiate local calcium spikes dependent on NMDAR that are highly localized to basal dendrites. So we focused on these neurons in our study. This is important here to mention and highlight that this work was led by two postdocs in my laboratory, Laura Maglio and Ricardo Gomez. And also in the work that I am showing today were highly involved David Bartolomé, Alberto González, and Belinda Rivero. And this project is still going on in the lab, and the team includes Andrea Reyes, Teresa Mínguez, and Rebecca that are exploring many other aspects that I will not have time to tell you about today. So in brain slices, Ricardo and Laura patched the layer 5 neurons and applied NMDA paths at basal dendrites, recording the resulting currents in magnesium-free medium, including glycine and TTX. In these conditions, we found two populations of neurons. About 60% of the neurons showed only inward currents activated by NMDA. We named them A-type neurons, and I will represent them from now on always in red. In contrast, about 40% of the neurons showed an outward current immediately after the inward component, very similar to what I just showed you that had been seen in other neuronal regions. We named them B-type neurons, and I will represent them from now on color blue. Importantly, this effect, this differentiation between these two neuronal types only occurred when basal dendrites were puffed with NMDA. Puffing NMDA at distal dendrites only produced inward currents, or at oblique, at other dendrite locations. Of note, there is the effect on the net NMDAR charge. B neurons, which are here the blue points, show significantly lower NMDAR net charge than A neurons, which are in red, at physiological potentials. Now, the other difference here is that when we calculate the ratio between the inward and the outward charge, then we see that that ratio is basically 0 in A-type because they don't have an outward component, whereas in B-type, the outward component is clearly present in and shown in this calculation. This will be relevant when we want to compare coupling in different conditions. So we logically suspected that these currents were driven by NMDAR and BK, and this idea was confirmed by pharmacology. So this graph shows the normalized inward currents, the bottom half, and the outward currents, the top half, recorded at -20 in the presence of different channel blockers. The inward current was completely inhibited by AP5, which is a specific inhibitor of NMDAR. These also abolished completely the outward current, indicating that the outward current is actually activated by calcium entering through NMDAR. Indeed, the outward component is BK, since it is completely blocked by paxillin. And blocking BK restores the inward current, here in dark blue. We additionally explored what were the GLUN2 subunits that were involved by using zinc and ifenprodin. Both inhibitors partially suppress the outward currents, indicating that both subunits could be involved. Of note here, we saw that if a protein quite consistently seems to produce larger reductions in the outward components, and this suggests that the complexes containing gluon 2B may contribute more effectively to the coupling mechanism. We have some data in the lab that seems to support these results, but I really don't have time today to tell you all about this. But this is a very interesting question that remains, and we are working hard to answer soon. So in the next set of experiments, we used calcium chelators to assess the distance between BK and NMDAR in these functional complexes in neurons. Briefly, the idea here is that we can use calcium chelators, BAPTA and ETTA, and they have similar affinities, but they show different association rates, and therefore they can be used as chemical rulers. And in fact, this is quite a conventional approach when working with association of calcium sources and calcium targets in nanodomains. In the presence of BAPTA 15 millimolar, which is the fastest chelator, no outward currents were observed in any neuron tested. Importantly, this additionally demonstrates that calcium entering through NMDAR is activating BK. Now, in the presence of ETTA 15 millimolar, some outward component was observed, but still the chelator was able to trap calcium entering via NMDAR before it reached BK. And this can be seen here, as I mentioned before, by seeing that this ratio, the inward-outward ratio, is increased, but not as much as we saw before in the neurons, in the B-type neurons. Finally, BAPTA 1 millimolar shows that the coupling shows a coupling that is basically indistinguishable from that recorded in the absence of chelators. Altogether, these results demonstrate that both channels are located in close proximity. And in fact, these kind of experiments can be used quantitatively to estimate distances. And the distances that we estimated were to be between 15 and 60 nanometers, forming what are called nanodomains. The data that I just showed you is all recorded from brain slices in actual native neurons. Now, in the lab, we have made a significant effort to set up an experimental model that allows us to study the NMDAR BK complexes in heterologous expression systems, such as hex cells. And this is very interesting because this allows us to answer different kinds of questions, first of all, but because it allows us to co-express different proportions of ion channels, it allows us to co-express with mutations that are disease-related, and obviously this broadens the capability of our experimental design. So we can co-express NMDAR and BK in membrane patches that we can excise from the cell. And then we can excise these to a bath that has no calcium. So calcium is only in the pipette. The only way that calcium reaches BK in the intracellular side to actually activate it is by entering through NMDAR. So we can estimate how NMDAR is involved or how this coupling is involved in activating BK. And this activation can also see by using this association can also be estimated by using other approaches. And the first one that we used is the proximity ligation assay. And very briefly, this system, and I apologize because it seems that in the screen it is not very clearly seen the signals, but believe me, they are there. Basically, how this works is that proteins that are very close one to the other, by using antibodies and system that uses slightly overlapping oligos and intercalating fluorescent nucleotides, then we can estimate protein-protein interactions. And these quantifications showed us that BK can associate with gluN1 2A and gluN1 2B NMDAR being expressed, as I mentioned before, in HEX cells. And also, we did electrophysiology experiments where we used this property of calcium shifting the GV curve of BK. In this case, this graph shows typical BK GV curves that are shifted by calcium, like the ones that I showed in my first slide. When NMDAR is co-expressed with BK, including calcium in the pipette, but in the absence of calcium in the bath, as I briefly mentioned before, a shift in the GV curve of BK is observed. Actually, this shift is equivalent to the shift that is produced by around 10 micromolar calcium in normal conditions. Interestingly, when these experiments are performed in the presence of physiological conditions, concentrations of sodium, the shifts were smaller, but still significant. Indeed, in these conditions, the shift produced by NMDAR containing gluN2B seemed more prominent. And we reasoned that this is due to the lower deactivation rates of gluN2B in physiological conditions. And this also matches the results that I showed using pharmacology. And as I said, more evidence that we are still gathering in our laboratory. Coming back to the somatosensory neurons, we wondered if there were other characteristics then that would allow us to distinguish type A from type B neurons, because there were no actual histological differences or any other differences that we could actually see when we were patching those neurons. So we recorded the action potentials in all neurons and then recorded the same neuron in voltage clamp to measure the basal dendritic currents and identify which ones were A and B and correlate that with their electrical activity. Interestingly, we found that the presence of NMDAR-BK complexes is restricted to a population of regular spiking neurons that are characterized by the absence of the so-called calcium spike that had been identified before in a subpopulation of layer 5 neurons from the barrel cortex. And this was known as the calcium spike. In our hands, and this has been really largely confirmed in the lab by now because we've done many experiments with this, the A-type neurons show this calcium spike, whereas the B-type neurons do not show that calcium spike. But other than that, every other parameters related to intrinsic excitability were identical between these two neuronal types. Now, the other question that we had was, okay, maybe the difference is that one neuronal type expresses BK and the other doesn't. And that certainly was not the difference between the two neuronal types because we saw that both of them expressed BK. So our working hypothesis is that somehow the B-type neurons direct BK to dendrites more efficiently, or maybe they express other proteins that are involved in forming the NMDAR-BK complexes more efficiently. We are currently working in the lab to identifying the proteins that form part of these multi-channel complexes. So far, then, we propose that NMDAR is associated to BK in postsynaptic sites. In neurons type B, a negative feedback mechanism would take place, where the calcium entering through NMDAR would activate BK channels, which in turn would produce driving potassium mounts, would produce a hyperpolarization that would restore the magnesium block and then blunt the NMDAR-mediated calcium signal. So if NMDAR BK complexes in the post-synapse were producing this effect, then this should have a significant impact, first of all, in synaptic transmission. And here, with this set of experiments, we tested this idea. So we electrically stimulated the afferent inputs that are reaching the basal dendrites and recorded the evoked postsynaptic potentials, or PSPs. It is important to note that this was done in the presence of magnesium. So in these experiments and the ones that I will show after these, NMDAR is functioning physiologically with occurrence of voltage-dependent magnesium block. So first, what we did was adjusting the electrical stimulation intensity to obtain PSPs between 3 and 5 millivolts. In these conditions, adding Paxilin produced an increase of the PSP amplitude and slowed their kinetics only in B-type neurons, indicating that the presence of BK in these neurons is indeed reducing NMDAR contribution to the PSP and therefore regulating synaptic transmission. Next, what we did was increasing the stimulation intensity to reproduce the NMDAR-dependent calcium spikes that are characteristics of layer 5 pyramidal neurons' basal dendrites. We blocked, in this case, the firing of action potentials by adding a sodium channel blocker in the pipette. In these experimental conditions, larger PSPs are observed as expected, and then addition of Paxilin in neurons type B resulted in an increase in PSP amplitude, very clear, with no effect on A-type neurons. Altogether, these results demonstrate that BK channels in these NMDAR-BK complexes can and do abrogate NMDAR currents, even if NMDAR spikes were taking place. So all the data that I have shown until now led us to hypothesize that there should be a significant difference in plasticity between neurons A and B. As a consequence of the reduction in the net NMDAR currents, calcium currents, caused by the close location of BK channels, B neurons should exhibit reduced plasticity. So what we did was we studied the effects of inducing spike time independent long-term potentiation, TLTP, in these neurons. Basically, what we did was electrical stimulation of the afferent inputs was used to evoke PSPs, and then pre and post associations were performed at different frequencies. And in these experiments, as I show you in a representative graph here, time-dependent increases in PSP amplitude, it corresponds to increased plasticity. So this would be representative of a neuron that is undergoing plasticity. Now, what happened in our case? So in our case, what happened was that a low number of pre-post associations of 30 pairings, as shown here in the figure, induced the long-term potentiation of postsynaptic potentials in A-type neurons, here in red, as supposed as known to be happen in these neurons, but then produced no effect on B-type neurons that are shown here in blue. This was really exciting, knowing that this was the proof, the actual proof that having BK in these complexes was basically blunting plasticity. Now, because this effect is totally consistent with this idea that BK is blunting the calcium signal that is driven by NMDAR. Now, if we add Paxilin and then we block BK, this effect was completely abolished. And basically, A and B type neurons were able to undergo plasticity. And then just a quick note here, these neurons were known to undergo NMDAR-dependent plasticity, and this effect, all the effects in all kinds of neurons, was blocked by AP5. And this was an important control to confirm that this plasticity, as had been previously demonstrated, is NMDAR dependent. And again, this effect is restricted to basal dendrites because this effect does not occur when apical afferents are stimulated. There was no difference and very little plasticity undergoing. Now, we have shown that BK reduces but does not completely abolish NMDAR currents. So we hypothesized that by tuning the experimental conditions, B cells may be able to undergo plasticity. And indeed, modifying the number of pre and post associations regulated the extent of TLTP, both in A-type neurons and in B-type neurons, although there was still a significant difference between A and B-type neurons. Interestingly, modification of pre and post-synaptic coincidence activity by increasing the number of pairings and the frequency modulated the potentiation extent, reaching similar levels in both neuron types A and B when extreme conditions of frequency and number of pairings was applied. So these data show that NMDAR BK associations confer B-type neurons with a fine-tuning mechanism of selective synaptic plasticity attenuation, allowing them to discriminate the amount and the frequency of synaptic inputs. And then this leads and brings me back to the very first slide that I showed, where the mechanism by which NMDAR with the calcium entering through NMDAR triggers these calmodulin kinase 2 signaling pathways that are involved in increasing the number of AMPA receptors at the membrane, and that is one mechanism that may undergo plasticity. Now, we are very tempted to propose that there may be another mechanism that should be added to these possible scenarios, physiological scenarios, where somehow, if the neuron regulates the number of BK channels that are associated to these NMDA receptors, then it could also regulate the plasticity. So creating the dynamics of these nanodomains may be another mechanism to actually regulate neuronal plasticity in these and other types of neurons. So all our data, oh, sorry, I don't know what has happened here. Oh, just going very slowly. So all our data then now proposes that the in neurons type A, what happens is that NMDAR is not associated to BK, and NMDAR here is shown in purple. So NMDAR is not associated to BK. These are A-type neurons. So calcium entering through NMDAR will trigger the regular pathways that are involved in plasticity in conditions where this is happening, and then it will lead to increased levels of AMPA receptors, and this synapse is going to be more receptive, and plasticity is going to happen in the conditions at which in that neuron type that happens. But in neurons, in this case, in neurons type B, in somatosensory cortex, but we have also seen that this happens in other regions of the brain. In B-type neurons, what happens is that associated to NMDA receptors are BK receptors, here shown in green. So what happens is that calcium entering through NMDAR is activating BK. BK drives potassium out of the cell. This leads to a hyperpolarization. The hyperpolarization restores magnesium block. And then this calcium signal is blunted. And therefore, is not sufficient, or to some extent does not reach the same extent as it was reached in the A type. And therefore, the level of increase of AMPA receptors here, we kind of propose a dramatic effect of not having any kind of AMPA receptor expression. But of course, one can modulate that response. And then what happens here is that this response is blunted, and then LTP is seriously compromised. We know that here we represent this because we know that this is BK mediated, because as you saw, it is this effect is blunted by the addition of paxillin, which is the BK channel blocker. So I hope during this talk, I have clearly convinced you that NMDA-RBK channel complexes play a physiological role in the synapse, that the association between these channels is important and constitute nanodomains because they are located in close proximity between 15 and 50 nanometers. This constitutes a mechanism of selective plasticity attenuation. And this is very relevant in the function of different circuits. And I have had no opportunity to tell you today about some work, unpublished work from the lab, where we now know that this association occurs in several brain regions other than the barrel cortex layer 5. And we also know that this functional association has pathological relevance, as is altered in different diseases where synaptic function needs to be or is compromised actually in the disease. And the most important slide here in this talk is the people that actually are doing the work. I already mentioned part of them. The team that is still in the lab are David Bartolomé, Rebecca and Teresa, Andrea y Belinda, that have done a lot of the work, part of the work that I showed, and a lot of work that I would show, I hope, in future opportunities. And of course, the work, this work could not have been done without Ricardo Gomez and Laura Maglio, who really drove the work that I showed today. And of course, Alberto González Hernández, all of them are continuing their scientific careers around the world. And then finally, we have a big network of collaborators that have collaborated with the lab at different points with very useful discussions and support in many ways. And of course, the funding from different sources that have confided and trusted us to continue doing our work here. And that's all I have. Thank you very much again for this invitation, and I will be very happy to answer any questions that you may have. Thank you.
Okay, thank you, Teresa. Thank you very much for this interesting presentation. And yeah, while I think we have to wait for some questions, so I will start with some curiosity. First, regarding the coupling, the physical coupling of the BK and the NMDA-AR, do you know if there is a direct interaction? Have you performed some immunoprecipitation assays or such kind of, because with the PLA, are you sure there is a physical, maybe this is not very important because you have clearly determined that there is a functional coupling, but do you know anything about this?
Yeah, that's a fantastic question. Yeah, well, we did not do a co-IP. We've done some now with another, trying to answer another questions, and they co-IP. BK and an MDAR. But personally, and this is an ongoing discussion, I don't think that co-IP necessarily shows direct interaction. If it is a complex where there are other proteins involved and the association is very tight, you could also see co-IP. As I say, this is kind of a more discussion about techniques and all that. So we do not know. But we are doing a couple of large assays in the lab where we will use actual proteomics like bioIDs, where we are going to assess the composition, the different proteins that are involved. We are sure that this is a macro complex. It is not just BK. And so there are probably other regulatory proteins, other proteins that are there. And we will see what it is, but so far we don't know what other proteins are involved in this association.
Okay, yeah, this was my other suggestion to try to see the proteomics profile of these membranes, and also for the lipidic composition of the membrane in these nanodomains. Do you have any idea of if it could have also a role in this coupling?
Yeah,
I suppose it's
that's an
difficult
excellent question, and we do not know.
Yeah.
We do not know. Honestly, I would expect so, but there's so little known about the membrane in composition in these nanodomains.
And well, let me see. I can't see any other question in the chat, so I'm going to. Alba Monesteiro. I don't know if she is going to make a question. Yeah, Alba, do you want to ask directly? If you can't write it in the chat, Alba Mosteiro. Maybe you can open your microphone and ask your question yourself.
In the meantime, I have one question. Okay. I'm really curious. I found it really interesting. Thank you, Teresa. I'm from another field, but well, I had lots of questions, and I see that this is really molecular work, really important. But going to diseases, I guess that there's no much, no? But thinking about muscular atrophies and all those kinds of dystrophies, and do you know something about these channels, how they work? Or are you, for example, able to isolate those type A neurons, type B neurons, and do a proteome and a genome of those neurons? Is there any way to do that? Or
well, one thing that can be done, I say can be done, I've heard.
There are like two questions. First of
obviously, yeah. Obviously, one could do something really fancy, which is that, I mean, there's no way for us to distinguish these neurons so far other than electrophysiologically. So what you can do is you can patch the cell, identify it as an A or a B type cell, and then sack the cytoplasm. And that is what is called patch-seq. So and then you can do RNA-seq with those cytoplasms.
Yeah.
And that is something that we are currently starting to set up in the lab, but obviously it's not easy. So we are going very slowly, and that is something that we would like to do. The interesting part here is that although I couldn't say more here, but this work, I'm sorry, in the lab we have seen, sorry, I was just looking at the chat and seeing some questions and I got distracted. So we've seen that this not only happens in somatosensory cortex, and in some other regions, it happens in all neurons.
Yeah.
So it can be even more interesting than what we think.
Yeah, I was
and
yeah, that's why I asked about the muscle, because since it's all irrigated and well, that maybe could also be all those channels in neurons in other parts of the body or
Okay,
exactly.
so we are receiving some questions in the chat. The first one is from Antonio Martinez Ruiz. He's asking if is there a redox regulation of system modifications of the interaction of these channels, and he also says that there are at least reports on S-nitrosylation of cysteine residues, and they could easily be extended to other cysteine oxidations.
That's a very good question. My short answer, this is so frustrating. My short answer is I don't know. We know that BK has a lot is regulated by post-translational modifications, and we are exploring part of them, also phosphorylation. And so far, it has been, what we got explored is there's no role. And there's some other work that is still in progress. So we don't know. This has opened more questions, as you can imagine, than hands that we have. But obviously, that's a very important question. And in the context of BK channels, is very relevant because these channels are known to undergo quite a lot of modifications that regulate their function in different tissues.
Thank you, Teresa. And then we have another question from Alejandro Samhan. First of all, he congratulates you for the nice work. And so he would like to ask if you know whether these nanodomains would reflect different pools of receptors that might be associated with cholesterol or not. Coming back to the lipid composition.
Back to the lipid composition. That's a key question. And of course, that is something that we have to address. And in fact, this idea of the cholesterol has been, actually, I really look forward to seeing that work, because there's a group in Seattle, Oscar Vivas, that are working on that related to BK clustering. And actually, they have it is not published yet, but I know that they are going to show it at the Biophysical Society meeting. So I am really eager to see what they have. So this is not something that we are directly working on, because it is impossible to cover everything. But it is an active and really heated question in the field.
Okay, thank you. Well, in the meanwhile, maybe there are some other people who are thinking about some questions. I was wondering, Teresa, regarding the pathophysiological part of the implications of your work. Do you have access to samples from these kind of diseases that you think that this coupling could be relevant? Because it could be nice to see if the proportion of A and B type neurons is altered in these, I don't know, these tissues.
Yeah.
Or, because I understand it's difficult to perform some biochemical analysis of the two types of neurons, but somehow to
well, we have the advantage that even though we cannot do, we can obviously, but it is more complicated to do these biochemical profiles and things like that. But electrophysiology is super cool. So with electrophysiology, we can actually assess if the functional coupling is altered or not. And I cannot disclose what we've seen, but I will tell you that there are a few pathologies where it is actually altered. So this is something that is now going on in the lab, and it is really nice because we are actually seeing 3 different pathological contexts where the association of BK and an MDAR is altered. and consistently. And we can see, so there are two things that we are seeing, which is that the proportion of A versus B type neurons is completely altered, but also in B type neurons, the coupling is much less efficient. So it is not only that the number of neurons, the proportion of neurons is altered, but it is also that the coupling is less effective for some reason. And we are using now super resolution microscopy to actually study how these complexes are altered in these kind of diseases. And what we see is that what it seems is that the pattern of BK versus an MDAR in these complexes is altered. So this contributes to less effective signaling of calcium towards BK from the entrance from an NMDAR. And thus it results in altered coupling, which may or may not, but certainly there is a clear correlation with these pathological conditions. So we really think that that it may be a mechanism that may be involved with the pathology.
That is really interesting to be able to correlate this pathology.
Yeah, of course. At the end, that is what really is the main proof of the physiological relevance, right? When you find a pathological context where this situation, where this mechanism is altered.
Okay, so thank you, Teresa. I don't know if I can't see any other question. Patricia, I don't know if you want to say anything else. So just
questions. Thank you very much, Teresa.
let me thank you again, Teresa,
Yes, thank you.
for accepting the invitation, and Patricia for organizing this, and all of you for attending.
Yes. Thank you very much.
Thank you very much.
Thank you. Bye.
Thank you.
Bye-bye.